Yes, my name is Heiko Lickert and I am the director of the Institute for Diabetes and Regeneration Research. Yeah, so the current treatments treat the symptoms, so basically the high blood sugar levels, but not the root cause. And that is also where I have dedicated my research life to basically find something, medication or a therapy, which can treat the underlying cause of the disease. And in our case, we're focusing here on the insulin-producing beta cell, which in type 1 diabetes gets lost through autoimmune destruction. And in type 2 diabetes, beta cells just fail because they have to see too much glucose and too many lipids. And that's when they cannot cope with that anymore, and then they fail too. So we try to basically fix this problem and have a curative or a causal treatment for diabetes. Yeah, we are part of the ISLET project, which is an EU project. We're also part in Germany of the German Diabetes Centre, where we have also activities around stem cell replacement therapy, we call it, or stem cell beta cell replacement therapy. But it's also a worldwide effort, one has to see it. So we are a little part of a big enterprise. So our focus is really the first important thing is we have to understand how pancreas development works across evolution, because very often we have model organisms in the laboratory. A lot we work with mice, but obviously human development is not the same. And so we have to understand how the pancreas develops. We have to understand how hormone-producing cells are formed in the pancreas, and we have to understand how alpha cells, which make glucagon, or beta cells, which make insulin, are formed during development, that we can then take these in vivo principles to translate them to the culture dish, where we then guide the pluripotent cells into the beta cell lineage. And there is still a lot of improvements to be done because the protocols are not super efficient. And we have to upscale then also that production. And upscaling means getting more cells, and the more efficient these protocols are, the cheaper it gets and the easier you can generate these cells. The other thing is, obviously, we are using the stem cell model to get cell replacement therapy going. And currently, the cell products which we generate are not exactly that what we have in our body. So first of all, we generate beta cells which are pretty similar to the beta cells we have in the body, but we still know they're not yet perfectly functionally mature. What does it mean? These cells do not respond perfectly well to nutrients and glucose in terms of their insulin secretory response. So there's still a lot of research to be done to get more mature beta cells. The other thing is we use the stem cell model also to identify novel drug targets. So there was a lot of effort done worldwide to find in type 2 and in type 1 diabetes risk genes. Now we know because both of the diseases have an underlying genetic cause. And if we know the risk genes which lead to diabetes, we in principle have therapeutic intervention points. So I make a simple example. We identified an insulin inhibitory receptor. We studied this in mice, and we saw that basically targeting this receptor, which means take the receptor out of beta cells, makes them better, makes them regenerating, makes them protective. But then we need to have drugs, and we need to have this human translation, because from mice we cannot directly go into clinical studies. And so here the stem cell derived beta cells, which are human stem cell derived beta cells, is the first way of translation, where we can have disease genes or potential therapeutic targets. We can show that these therapeutic targets are valid targets, and we can develop drugs against these targets to have the same effects as we have when we knock out the genes. So we have now a therapeutic intervention in the cell culture dish, and then we can follow or translate this towards clinical testing. I think all what we do is important. So I should probably say our big aim is to either replace insulin-producing beta cells by the activities we do in the ISLET project for cell replacement. But there is another way of thinking. If you have type 1 diabetes and you could stop the autoimmune attack and you could basically get the beta cells back into function early in the disease progression after the patient come to the doctor and the disease is diagnosed, there's still some remaining beta cell function. So if you stop the autoimmune attack and convince the beta cells to get back into function, that would be a different approach. That would be an approach which you would do by pharmacological intervention, and more people probably could benefit from that approach in comparison to an organ transplantation, a cell transplantation, or a stem cell beta cell transplantation, which currently also comes with what we said before, that you require immune suppression, and we need this solution how we can have a transplant without a lot of immune suppression, maybe with a mild immune suppressive regime, which could be tolerated over a lifetime. In principle, the stem cell therapy can work, but the problem with it is currently that it's too expensive, that you need immune suppression, and we cannot talk about a cure for diabetes. We can talk about normalization of blood glucose control, but we cannot talk about a cure, because basically you're changing a cell product, a stem cell derived beta cell product. You don't need insulin anymore, but now you need immune suppression. So if we would be able to modify the cells, with gene therapy comes now into play, in a way that they could evade the immune system, so that they are not detected as foreign and also not destroyed, like what the problem is in type 1 diabetes, the autoimmune attack. So the immune system attacks their own cells. So if we can protect them from this immune destruction, I think this therapy could be used in a much wider and bigger patient cohort. And in 10 years from now, I really hope that we have a combination out of a stem cell product and an immune strategy to reach a larger patient group. And I think it will get accessible for for people living with type 1 diabetes, which really require an islet transplant.